So here's another video tutorial in my series of tutorials on metabolism. We've looked at glycolysis before. We've looked at the matrix reactions. We've looked at electron transport system and fermentations. And so far, all of those have pretty much dealt with carbohydrates entering the metabolic pathways of aerobic cellular respiration or anaerobic respiration when glycolysis breaks off into fermentation. We haven't really looked at the entry of other food types. So let's say you're eating a hamburger or something. You're definitely going to get some starch, some sugars in there. But there's also protein and there's also fats. And of course, there's nucleic acids, but in this tutorial, I'm not going to be looking at that. So this is for grade 12 biology, perhaps a little bit of a review for first year university, although I'm sure you'd go into much more detail. The image that you see here is a worksheet of mine that you can print out. It's on my website at www.zerobio.com down here. All right. But I'll also put a direct link to the worksheet if you want to print it off and follow along with this video. And you don't want to go all the way over to my website, right? Why wouldn't you want to do that, though? Okay. So let's have a look at metabolism of food. We're going to start actually here in the middle, and we're going to actually look at carbohydrates first of all. So if you've got the printout and you want to color it as you do this, or at least write the stuff in the boxes, you could do that. So here I've got what would represent a number of glucose molecules in a chain. So this could represent some form of polysaccharide. So we're going to write that in the box there. This could be starch, glycogen, something along those lines. So we've got a polymer of glucose units all joined together there to make up a polysaccharide. So it's a sort of a complex carbohydrate. And these are going to be broken down by various different enzymes, but in this little spot here, this is where we're going to put the enzymes. So we're going to put an enzyme there, and here, and over here. I know it's a little small to write in, but this is carbohydrates. And that could represent amylase or some other carbohydrate splitting enzyme. So I'm just using the general name, the general categories of the enzymes and not getting specific. So when the carbohydrate enzyme acts and breaks down the polysaccharide, we could break it down into simpler sugars. All right, so simple sugars goes there. So for example, I'm illustrating here a disaccharide like maltose or sucrose or lactose or something like that. All right, so we go from polysaccharides to simple sugars, simpler sugars anyhow, using the carbohydrate enzyme, and then something that will split into until we get the individual glucoses. Now, the first step of glycolysis is glucose, but I've generally put glucose 6-phosphate here. I'm following roughly another textbook that begins with that, because once the glucose is in the cell, it's going to very quickly be phosphorylated to glucose 6-phosphate, and this is going to give it an extra charge that's going to make it more difficult for glucose to leave the cell. Because of that extra charge, it sort of bounces off the cell membrane and it can't escape. So glucose is now confined to the cell. Now, this is glycolysis here, but I don't have all the steps because I just want to show where various food types feed into the pathway. So as we move along the steps of glycolysis, glucose 6-phosphate, fructose 6, fructose 1,6, etc., we eventually get down to G3P or glyceraldehyde 3-phosphate. That will eventually be converted into pyruvate, and that's our last step of glycolysis, right? So we're not dealing with all the intermediate steps here. We're kind of summarizing glycolysis. And as you know, pyruvate will then enter the matrix of the mitochondrion and be converted into acetyl-CoA. So these are the basic steps for carbohydrate metabolism. The acetyl-CoA will then go into Krebs cycle. Okay, so make sure you have all those labels, and you can see that's the central pathway, essentially the preferred pathway for cells to obtain energy. Okay, now, you're eating your hamburger or whatever, and you're getting your polysaccharides, and this is what's going to happen. And of course, ATP is going to be produced both in glycolysis and in Krebs cycle. NADH and FADH2, the electron carriers, are going to be produced. They're going to go over to the ETS. So we're not showing that here, but we know that our goal is to make ATP. Well, let's have a look at where do we want to go, right or left? Let's go with the right. Let's go with lipids. So you get some fats in there with your food, with your hamburger, let's say. And although there are different types of lipids, I'm going to focus on triglycerides. So I've got images here for representing triglycerides with this box and the three little rectangles coming down, and that's going to represent the building blocks of triglycerides. Now, lipids can be broken down by a category of molecules called lipases. All right, so we've got carbohydrate, ASE enzyme, that breaks down the carbohydrates or polysaccharides, at least in the first step. And we've got lipase that breaks down lipids. Triglycerides are made of two basic building blocks. All right, we're going to get fatty acids and we're going to get glycerol. Now, of course, that's for one triglyceride, but imagine a fat or even some lipid, you're going to have lots of those triglycerides, right? So we're just kind of simplifying things here. Now, glycerol is a 3-carbon molecule, and we have seen it before in a previous video, I'm pretty sure, but this is the molecular structure. This is at least the way I'm drawing it here. And if you wanted, you could color these in. Carbon is usually black. I'm talking about the typical molecular model colors, you know, if you build this stuff in a lab. Oxygen is usually red. Hydrogen is usually white, but against a white background, I just wanted to add a bit of color, so I've done it in a sort of a light gray color. But this is a molecule of glycerol. And glycerol is very close in structure to glyceraldehyde 3-phosphate. And so when your lipids are broken down by lipase into glycerol and fatty acids, the glycerol molecules can enter the glycolytic pathway by being converted into glyceraldehyde 3-phosphate. And we're not worrying about the specific enzymes involved. But once the glycerol goes in, now it can continue with the steps, be converted to pyruvate, etc., and move down, and any NADHs or ATPs or FADH2s can be produced, you know, from then on. It doesn't matter that it's skipped a couple of steps up here, as long as it gets into the pathway somewhere and continues. Now, fatty acids, as you possibly know, have a carboxyl group at one end, and I've drawn it as C double bond OH. I've left the H on, although in solution, a lot of these H's are usually gone, and there's a minus sign. They're ionized, and if this was drawn in the ionized form of just C double bond O and O, and the H wasn't there, and there'd be a minus, you could probably imagine that as CO2. So fatty acids can be decarboxylated. We looked at that in fermentation to release a CO2 molecule that you can just exhale. And then what's left is this long carbon and hydrogen tail. Now, I just drew this with 1, 2, 3, 4, 5, 6 carbons, all right? But many fatty acids are very long carbohydrate or hydrocarbon, carbon and hydrogen tails. So here's some colors. Again, black for carbon, red for oxygen, and light gray for hydrogens in my case. So imagine that this fatty acid is cut into 2 carbon units. Let me highlight that. Okay, so imagine an enzyme comes down and cuts it there, and cuts it there, and cuts it there, and takes that, and you exhale that as CO2. Well, these molecules are very similar to acetyl-CoA, 2 carbon molecules, a slight, you know, rearrangement, and that has to go on. So fatty acids are converted into acetyl-CoA. And you get lots of acetyl-CoA molecules from one fatty acid. Well, imagine a triglyceride with 3, all right? And then imagine a lipid, again, with, you know, thousands of triglycerides. So think of all the acetyl-CoAs that can feed into the metabolic pathway there. So admittedly, you miss producing what? 2 net ATP? You know, who cares about that? And 2 NADHs, you've now got acetyl-CoA, and lots of them. And they will then go down through Krebs cycle. You'll make your bunch of NADHs and FADH2s, which will go off to the ETS to produce lots of ATP. So fats store a lot of energy. There's a lot of potential to make ATP from fats. Okay, so that's how lipids enter the biochemical or metabolic pathway. Glycerol at glyceraldehyde 3-phosphate, for the most part anyhow. Fatty acids at acetyl-CoA. So there's our carbohydrates and our lipids. The other major macronutrient is proteins. We're not going to deal with nucleic acids. They're not really macronutrients anyhow. So if we come over here to the proteins, right? Proteins are made of amino acids. So this is my symbol for amino acid, just a chain of amino acids here. All right. But imagine a typical protein is actually hundreds of amino acids, not 4 as I've drawn. All right. Now, you need protein to build your antibodies, your enzymes, some hormones, your muscles and bones and body, organs and stuff like that. So protein is not a primary energy source, but it can be used for energy. If you're eating protein and you're building body stuff and your body goes, okay, I've got enough muscle, I've got enough antibodies for now, thank you. And maybe I'm lacking a little bit of some of these other ones, you can use proteins. Proteins are broken down by many different enzymes, and there's many different types of proteins. You've probably heard of pepsin before, and trypsin, or repsin perhaps. So I'm just calling these protease, all right, as a general name for protein degrading enzymes. So proteins are broken down by proteases eventually into amino acids. Now you may go down to polypeptides and peptides and dipeptides, but eventually you're going to get the individual amino acids, and there's my symbol for a single amino acid. Now, amino acids are a little bit different, because amino acids, as you know, have an amino group and a carboxyl group, all right? We're just going to focus on the amino group here, and an amino group is NH2. So I'm writing NH2 in that little box there. Now, because nitrogen normally forms 3 bonds, not 2 like this, there's some lacking right here. There's some excess electrons, and this generally tends to attract, let me draw this in here, just so I can use a pen. It generally attracts another hydrogen atom here, which would attach there. And that would produce that's ammonia right there. And ammonia is toxic. These amino groups have to be removed from the body. And so the amino groups are going to be clicked together to make this structure. So you've got an amino group here from one amino acid, an amino group there from another amino acid, and it's going to be clicked together to make this structure. I'm using the same colors for carbon, oxygen, and hydrogen, but I'm putting nitrogen in here as blue to sort of match this color scheme. All right. Nitrogen may not be illustrated as well. Actually, it is rather light blue usually in bottle kits I've seen anyhow. All right. This molecule here is called urea. And it is a way for the body to take the amino groups and get them out of the body because they can be toxic. So amino groups are clicked together like this with carbon and oxygen, and that forms a molecule called urea. It may look to you a little bit like formaldehyde, right? Do you remember C double bond O and H on one side and an H, right? And in my class in grade 12 biology, we looked at the fact that formaldehyde is definitely a polar molecule. Well, so is urea. Nitrogen is charged, the oxygen is charged, that kind of thing. So, or at least partially charged or electron dense. So this molecule is very water soluble, and this is why this is one of the major components that will come out in your urine. Okay, and look at the similarities of the words urea, urine, that kind of thing. So that's how we get rid of our amino acids. This process of removing the amino group from amino acids, this is called deamination. The removal of the carbon dioxide is called decarboxylation. This is deamination. So that's how we will get rid of our amino groups. And what's left is the carbon backbone. All right, you've got the central carbon, you've got the R group, which could have lots of different carbon things. And that's why you see a lot of different arrows coming in here, feeding in at different spots. You can convert some amino acid carbon backbones into pyruvate and enter there, some to acetyl-CoA, some to various intermediates right within Krebs cycle. I mean, just look at the structure of Krebs cycle molecules and try to imagine that piece, I mean, they're only 4, 5, 6 carbons, in size, and think of the R groups of amino acids and imagine an enzyme splitting it into one of those Krebs cycle molecules and then that just feeding in. All right. So that's a general summary of how the major 3 macronutrients enter metabolism. Our central pathway here, which of course would have fermentation coming off of the side if it was an anaerobic environment. All right, our central carbohydrate pathway, and then the entry of lipids. And we haven't looked at every type of lipid, but just generalities, all right? The entry of lipids and the entry of proteins. So when you eat your meal, you've got carbohydrates in there, you've got lipids, you've got proteins. All of those can enter metabolic pathway to generate energy. And that's what this video was all about. So hope that helped you. See you again soon.
So follow the trail, little white diamonds that fell from your veil, deep in the darkness of ambery wood. But the devil stepped in by the light of the moon, saw a sword and a grin, and a beautiful girl on her knees where it stood. Ooh, Lord, first a twitch of the eye, then a thundering burst from the thigh. One shot and the devil, he bought himself one hell of a hole. There's pixie dust in the air, the smell of the sea and strawberry hair. I'm riding away and she's sailing my soul. You're secretly sailing, I'm secretly waiting for the night you're arranging to hold me within. A fire is raging, my spirit ablazing. The soul that you're saving is caught in a spin, caught in a spin.